Good morning, everyone from California, here where it's 6 o'clock a.m., 6:01. We wanted to give a brief review of what was covered yesterday and then an introduction for what's going to happen today. So yesterday there was a very nice session on problems with fat that was led by Russ Tracy. And Jordan Lake spoke on antiretroviral associated issues with fat and muscle density, and the very important topic of weight gain and how that's influenced by aging. Giada Sebastiani spoke on fatty liver, another very common issue and aging associated issue in people with HIV. And then there were several very nice abstract-driven sessions. Abstracts have been very strong this year. Joe Margolick led that session. And then in the afternoon, we had a couple of very nice presentations on the skeletal system on bone, one by Michael Yin from Columbia, focused on the bone and screening in the clinic and changes in therapy with tenofovir disoproxil fumarate and tenofovir alafenamide. And then Todd Brown returning to the HIV and Aging workshop, which we're all very pleased to see, talking about falls and fractures. Today we have a very nice program with very strong speakers this morning on neurology. And then some nice, a nice focus on sensory diseases with Douglas Jabs from Johns Hopkins and my colleague Ron Ellis from UCSD in San Diego, not San Francisco. And then in the afternoon, your time in most places, or early evening, still in the morning on the West Coast here in the US, Christine Erlinson, one of our co-chairs, giving some overview information, and then Marta Bofito talking about HIV over 50 clinic in London. Marta is a very accomplished physician and scientist who focuses on pharmacology, along with Meredith Green, Sarah Schmaltzley, and Ginny Sigler from New York. So it's really going to be a very nice day. And I'll go ahead and go right into then the introductions. For session 4 here on neurology. So very pleased to introduce Abi Nath. I've known Dr. Nath my entire career, and he is a very distinguished scientist in our field. And he trained not only in India, but in Texas and in Manitoba, and has held positions at Kentucky, faculty positions in Manitoba and Kentucky and Johns Hopkins before coming back to NINDS, where he's the clinical director and the director of the Translational Neuroscience Center and chief of the section of infections of the nervous system. So Dr. Nath has truly been made impactful contributions to the field, truly been a friend and teacher to me, and I'm very pleased he agreed to present today on HIV and Alzheimer's disease. So welcome, Dr. Nath.
Thank you for the kind introduction. Today I'm going to talk about the effects of HIV on aging of the brain. It's been known since the early days of the HIV pandemic that older individuals have more difficulty with cognitive function. And as shown here from work done by the CDC in the early 90s and late 80s, that those individuals at greater than 60 years of age have a rapid decline in cognitive function. And now, even in the antiretroviral era, whereby frank dementia is not that often seen, greater than 30% of individuals, even if their CD4 cell counts are under decent control, they still have cognitive difficulties. And just adding more antiretroviral drugs does not seem to solve the problem. And several studies confirm this now, and it's shown here on the left. You can see that even individuals who are less than 60 years of age have a more rapid decline in cognitive function. And here you can again see that older individuals who have HIV infection have more complaints with regards to their activities of daily living. And if you look at the type of dysfunction in these patients, you will see that these patients on antiretroviral drugs more often have problems with executive function, as you can see here, and they have more difficulty with learning. And so it's not all cognitive domains, but some domains are much more affected than others. One might argue that what kinds of effects that really has on a patient's functioning and long-term survival, and it's shown here that just by the fact that those individuals who have cognitive dysfunction tend to die faster. And that is because if you can't remember to take your medications, and if your cognitive function is impaired, you can't protect yourself in many different ways. And so a variety of different reasons can contribute to early demise in these patients. We looked at patients by age, categorized them into normal controls, those with asymptomatic impairment, mild, and those with frank dementia, and looked at neurofilament levels in these patients. And what we found was that the neurofilament levels go up with age. And also protein carbonyls, which is a sign of oxidative stress, also seem to increase with age in this patient population. And so, and these are individuals with HIV infection. However, the PET scan studies have not been all that conclusive. It's been hard to demonstrate a clear difference in the HIV positive patients compared to controls for increased uptake of the marker that binds to amyloid, showing that there's increased amyloid deposition. However, in some areas that might be the case. For example, here in the cingulate gyrus here, and you can see in the prefrontal area, there seems to be more deposition of amyloid than in other areas of the brain. Pathological studies consistently show that patients with HIV infection have more deposition of amyloid plaques. And so you can see that even at this age of 30 to 39 age group, these patients with HIV infection had more amyloid plaques in the brain compared to healthy controls. And at every age group, that seems to be the case. And here in another paper, they've shown the shape of the plaque. And you can see here, they call them diffuse plaques. So in HIV-infected individuals, you can see these plaques, but the borders of them seem to be a bit diffuse. And while in Alzheimer's plaque, you can see very crisp borders over here. But if you quantitate these number of plaques, you will see that they keep going up with the length of infection. Then how does HIV really lead to these plaque formation? And one way is to try to understand where HIV itself is being produced in the brain, and then what the mechanisms might be for interaction between the two. So HIV is a complicated genome, and it has it's a retrovirus, so it gets integrated into the DNA, and then a number of different genes are formed by splicing of the RNA. And so one of the first proteins to be formed is Tat, and it's formed off of 2 exons. And so if you look at by immunostaining in the brain, you'll find that Tat is present here in the perivascular cells, and these are all macrophages here, all this brown staining. And you can also see it in these microglial nodules. Okay. Here is staining for GP120, which is the envelope protein. And you can see these multinucleated giant cells staining brown, showing that there's increased production of HIV in these cells. You can also find HIV in astrocytes, and here you can see immunostaining for the NEF protein, and in situ hybridization showing the RNA for the GAG pol transcript in astrocytes. However, even though HIV is present in astrocytes and in macrophages, the cells that degenerate are the neurons, even though the neurons themselves are not infected. So you can see here that in the dentate gyrus, this is what the normal dentate gyrus should look like. This is a patient with no encephalitis. This is one with drug abuse. You can see that there's decreased staining. And if you look at this photomicrograph here, this is what the normal neuronal population should look like. And an HIV patient with dementia here, you can see there's profound loss of neuronal staining. Now, if you were to take a transgenic mouse in which there is made with these 2 genes, amyloid precursor protein and presenilin-1, they are at increased propensity to develop amyloid deposition, and cross them with the GP120 mouse, you will find that in these crossed animals, there are these huge plaques that form here of amyloid deposition. And you can quantify them, and you can see that in different regions of the brain, starting from the bregma and going posteriorly, you will see that there are huge amounts of plaques in the triple transgenic animals. And the size of the plaques is also greater in these animals. So the number of plaques as well as the size of the plaques. And now I'm going to show you that not just only GP120, but even the Tat protein can interact with amyloid as well in a very unique manner. So the Tat protein, as I mentioned earlier, can be found in these perivascular cells, but is also present as deposits in the extracellular space here. And it tends to aggregate very easily. So you can find these aggregates of Tat protein in this microglial nodule here. And you can also find Tat in the spinal fluid of patients who have been adequately treated with antiretroviral drugs for a long period of time. So this is an ELISA for Tat, and you can see that there is increased Tat that can be found in the spinal fluid here. You can also find it in exosomes from the spinal fluid. And the presence of Tat has consequences, because those individuals who have Tat detectable in the spinal fluid also had difficulty with information processing, and psychomotor domain was also diminished in these patients. And interestingly, we found that there was a propensity to be present more so in women and individuals who are actually even better controlled on antiretroviral drugs than others. And so it shows that just control and recovery of CD4 cell count alone is not sufficient to prevent its production. And that there's an inverse correlation in the presence of Tat RNA and HIV RNA and Tat, suggesting that once the virus is controlled, the proviral DNA is probably still trying to crank out Tat and trying, because it's so critical for HIV replication. So there is this positive feedback loop that leads to increased production of TAT in the presence of antiretroviral drugs. But it has multiple consequences. TAT can act on T cells and it can activate T cells. So you find a lot of activated T cells in these patients. The other thing it can do is it can directly interact with neurons and cause excitotoxicity. So as shown here, you can see that when you put TAT on these hippocampal slices here, they just start firing. And you can see that each line here is an firing from these neurons, and it just fires like crazy all the way through. If it acts on glial cells, it can lead to production of cytokines. So it can activate glial cells, and you can see IL-1, IL-6 being increased in a dose-dependent manner. And it also synergizes with other viral proteins, for example, with GP120 or glutamate, in order to cause more excitotoxicity. So here's a cell that is repeatedly treated with glutamate, and you can see the amount of calcium changes inside the cell decrease. However, if you pre-treat the cell with TAT and now put glutamate on there, you will see that the cell can no longer handle the calcium, and there's huge increases in calcium here. Similarly here, with subthreshold levels of GP120 and TAT put together, you see increased levels of calcium within the cell. And in transgenic mice here, if you put TAT and APP together, you'll see that there are these huge plaques that will form of amyloid beta peptide plaques here. And you will see that if you put them in culture, you will also notice that the neurons themselves start degenerating. So here's amyloid beta peptide with TAT, and we counted the number of puncta, which is an indication of neurotoxicity, and they go up in a dose-dependent manner. And if you look at the interactions of TAT and amyloid beta peptide by microscopy, what you will see is that here's the amyloid fiber, and these little bumps on top are the TAT sitting on there. So they decorate the fiber really nicely in a systematic manner, and ultimately they form lots of different types of plaques. And that's shown here diagrammatically. What TAT will do is just wrap these amyloid plaques together, and ultimately they form these huge plaques here. And these will then sit on a neuron and lead to toxicity of the neuron by making pores in the cell membrane. If you were to take TAT and put it in different regions of the hippocampus, you'll find that there's some parts that are more vulnerable than others. So the dentate gyrus here and the CA3 region are much more susceptible than other parts of the brain. So the question is now, can we block the effects of TAT? And the answer is there are many different ways of being able to do it, although none of them are currently therapeutically available. So this is beautiful work done by Susanna Valenti, and she's shown in a series of papers that this drug called cortistatin A can inhibit TAT, but once it does so, it will just lock HIV completely. So no other transcripts will be formed. So that's called a block and lock type of phenomenon. And because TAT is also responsible for chromosomal rearrangement, so once it's the chromosome is open and TAT is available, if you were to be able to block it, then it'll just lock the entire HIV genome right there for extended periods of time. And in our lab, we have made antisense molecules against TAT in order to be able to prevent the protein from being formed. And so these antisense molecules are strands of modified DNA and a combination of DNA and RNA moieties here, and they will then bind to the messenger RNA and lock it. and prevent it from forming protein at all. And we've shown here in these astrocytes where TAT is being expressed, and these are a panel of different types of antisense molecules, and we've identified some that act really well compared to others. So in summary, there is an acceleration of neurocognitive deficits with aging. There's uncontrolled TAT production and deposition of amyloid in the brain, and despite use of antiretroviral drugs. And a lock and block strategy with TAT antagonists can potentially provide a functional cure and stop age-related progression. So I'd like to acknowledge a number of people who have contributed to this. A number of them are not on this list, and I apologize for that. But people who developed the TAT assay was Carol Anderson, Muzna, and then Tori Johnson in my lab. All the work with TAT amyloid interactions done predominantly by Alina Hadigan, and the therapeutics work is being done by Lisa Henderson, who is leading the efforts. Thank you.
Hi, everyone. So thank you very much, Dr. Nath, for that really nice presentation. It's now, I just want to remind everyone that we'll have question and answer session after this next talk. It's a pleasure to introduce Dr. Angela R. Kammer. Dr. Kammer trained initially in Cluj-Napoca in Western Romania. We work in, I've done some work in Bucharest, and I've had the privilege of visiting Sibiu, which is just a few hours from Cluj-Napoca in the Carpathians. It's a beautiful part of the world. But she's been in the, she trained also at the State University of New York in Buffalo, also an excellent institution, have a trainee there as well. And she is, has been at the NYU College of Dentistry and is a research scientist in Brain Health Imaging Institute, the Weill Cornell Medical Center. She's going to talk today about a very timely topic. There's a great interest in the microbiome, but most work is focused on the gut microbiome, but the oral microbiome and the vaginal microbiome are also very important, and particularly in our patient population, because they don't always, our patients don't always have access to dental care. And so they can have pretty bad periodontal disease. And this can be made worse by use of certain drugs like methamphetamine as well. So it's a very, very timely topic. And Dr. Kammer is going to talk about oral health with aging and periodontal disease and the association with dementia. So, welcome, Dr. Kammer.
Good morning. I would like to thank the organizer of this meeting for the opportunity to present my data on periodontal disease and Alzheimer's disease dementia. One of the hypotheses put forward to explain the Alzheimer's disease pathogenesis is inflammatory pathogen hypothesis. According to this hypothesis, there is inflammation in the brain, which characterized by elevated markers, elevated molecules of interleukins, TNF-alpha, C-reactive protein. These pro-inflammatory markers have double role. One, to activate the glial cells to produce Alzheimer's disease specific molecules and also neurodegeneration. The corollary of this hypothesis is that any peripheral inflammation could potentially contribute to the pathogenesis of Alzheimer's disease. One of these conditions is periodontal disease. Periodontal disease is a peripheral chronic inflammatory disorders that result in the destructions of the tissue supporting the teeth. 64% of adults more than 65 of age have moderate to severe periodontal disease. But also periodontal disease is present in children, yes, in a lower percentage, and some percentage of children and adolescents, young adults have even aggressive severe periodontal disease, and also a high percentage of Down syndrome have aggressive periodontitis. Here in the upper picture, I would like to show you a normal periodontium. You can appreciate that sharp contour, sharp features, and the lack of inflammation of erythema and edema. And also you appreciate the lack of destruction of the tissue surrounding the tooth that it's exemplified here. In the lower picture, you see a periodontal disease picture from a patient. And you can see increased in inflammation as increased erythema, as you can see, edema, tooth mobility. And if you look in this picture, there is destructions of tissue surrounding the teeth. This destruction can be measured by pocket depth from here, from gingival margin to the depth of the pocket, or through clinical attachment level, which is just from the CJ, from the crown, when the root and the crown meet each other, to the depth of the pocket. Periodontal disease is a polymicrobial dysbiotic disorder. Bacteria is required, but not sufficient to induce periodontal disease. No one bacteria can cause periodontal disease. In 1998, Heffergy and Sokransky, very well known microbiologist, oral microbiologist, put together clusters, colored clusters, representing bacteria that associate with each other and associate with periodontal disease. And as you can see, there is here red cluster and the orange cluster. Because of their association with periodontal disease, they were called pathogenic cluster. And I will come back to this red cluster. This is an HIV meeting, so I assume people, most of them know about bacterial dysbiosis. But just in case, briefly, simply define, is a higher ratio of bad bacteria and good bacteria. But when we talk about periodontal disease, we talk about bad bacteria as bacteria that associate with periodontal disease on this column, on the left column, and good bacteria, bacteria that associates with healthy periodontium, and it's called good bacteria. I would like to illustrate here the effect of periodontal treatment on periodontal disease. Here you have a patient, and here is X-ray. And you can see here that the gingival margin, this is the gingival margin, which on the X-ray would be here. And to the bone crest, there is a lot of space. And this space is filled with inflammatory tissue. Here, after treatment, as you can see, the edema and erythema, as you can see here, lessen. And even on X-ray, we see a well definition of the bone crest, which indicates stabilization of periodontal condition. This slide represents a periodontal pocket. Here is the root, And here is the pocket that I showed you before on the other slide. And this is the epithelium that lines periodontal pocket. And you can see it here in this space. There is a lot of bacteria, bacterial products, and inflammatory molecules. But also you see that the epithelium here, the lining is ulcerated, causing the bacteria, allowing the bacterial products to get access to the systemic circulation, as you can see here. And here you see a lot of polymorphonuclears indicative of inflammation. Why is that important, that inflammation and bacteria that are getting into the systemic circulation? It's because they can induce systemic circulation. Based on longitudinal large studies, American Heart Association issued levels of C-reactive proteins and its level association with cardiovascular disease. Those levels were 3, low risk, intermediate, and high risk. And as you can see here, based on a meta-analysis, periodontal disease induced C-reactive proteins in the plasma that was in intermediate, high, low, high risk. And following periodontal treatment, that C-reactive protein decreased to lower levels. Based on inflammatory and dysbiotic burden, periodontal disease has been associated with a multiple of conditions: diabetes, atherosclerosis, chronic kidney disease, pneumonia, rheumatoid arthritis, cancer, and finally Alzheimer's disease. This is a model to illustrate the potential role of periodontal disease in Alzheimer's disease pathogenesis and pathology. And as you can see, bacterial products, cytokines, get access to the circulation and eventually to the brain, inducing the production of amyloid and of tangles. I would like to illustrate the data in the literature that shows that periodontal disease, inflammation and dysbiosis should be taken seriously as they associate with different measures of Alzheimer's disease pathology. Animal studies, a lot of them, using P. gingivalis, LPS from P. gingivalis, and other models of periodontal disease showed showed inductions of neuroinflammation, brain amyloid, neurodegeneration, cognitive dysfunction, even brain infection, or what I would call periodontal bacterial translocation in the brain. And also it showed that inhibition of virulence of LTL could reverse some of the effects. Epidemiological studies showed increased risk of dementia, Alzheimer's disease, increased risk of MCI, and increased risk of cognitive decline. Similarly, immunological studies looking at the IgG against periodontal bacteria, they showed increased risk association with increased risk of dementia and cognitive decline. And finally, pathological studies showed the presence of bacteria in the brain, and those are spirochetes and P. gingivalis. In my collaboration with Cornell Med, I started my collaboration with Moni De Leon from Cornell, and we have, until now, we have a pool of 75 subjects. What is important about this cohort, this pool of patients, is that they are cognitively normal, they are elderly, relatively elderly, well-educated, and relatively healthy. So it's quite a homogeneous group of people, and I believe the differences that we had in our data in part is because the population is homogeneous. We started looking at the association between periodontal disease and brain amyloidosis in the brain accumulation, brain amyloid accumulation. And we used attachment loss of an index of periodontal disease. Remember this periodontal destruction. And PIB PET, which is Pittsburgh compound PET, retention in the brain as an index of brain amyloid accumulation. And what we found was a correlation, high correlation. And here you can see a patient with high clinical attachment loss and with low clinical attachment loss, and you can appreciate the light up of the frontal and prefrontal areas. Next, we wanted to investigate the relationship between periodontal dysbiotic in subgingival environment and markers of Alzheimer's disease pathogenesis. Subgingival environment means under the gum line, and there is where the periodontal bacteria exists, and obviously periodontal health bacteria is. That's why we are using subgingival bacteria from subgingival environment. We assessed the bacterial abundance using 16S RNA amplification and and sequencing. Here, from the literature, we developed a Biotic Index, which was defined as a ratio of bad bacteria over good bacteria. And also we classify people with CSF A-beta 42 into two groups. One group that we called amyloidosis positive group that have low A-beta 42, and amyloidosis positive that have high A-beta 42. And we can see that this Biotic Index was significantly elevated in the group, in the amyloidosis group. Next, we wanted to investigate subgingival bacterial differences between A-beta positive and A-beta negative subjects at different ranking, bacterial ranking. And we did at phylum level. I don't show the slide, I don't show the data, but there are bacteria that are differences between the two groups. This slide shows the differences at the genus level. And as you can see, Bacterium, Provotella, and Dialister are increased while Corynebacterium, Actinomyces, and Capnocytophaga are decreased in amyloid positive group. To detect the differences, that biomarkers which would differentiate between amyloid positive and amyloid negative group, we used LEFSI, linear discriminative analysis effect size, at the species level. And as you can see here in the green, at A beta positive group, you will see species like Provotella species, Porphyromonas species, fried bacterium species. These are species that are associated with periodontal disease. While in a beta negative group, you can see here in red, Capnocytophaga, Corynebacterium, Actinomyces species. These are species that are prevalent in associate with periodontal health. To better exemplify the data that I presented, here I present two histograms, one from a healthy bacteria, Corynebacterium, and one from a bad bacteria, Bacterium fastidiosum. Each line here in red represents the abundance of the bacteria in one subject. You can also appreciate the pattern of lower abundance in the A beta positive of good bacteria and high abundance in A beta positive of bad bacterium, namely Bacterium fastidiosum. As I mentioned in animal studies and even in some clinical studies, P. gingivalis received a lot of attention because it's part of the red cluster as I described, but also it considered an important pathogen. And so I wanted to see if the same pattern exists in P. gingivalis. We can see a similar pattern, but it was not statistically significant. Maybe in a larger sample size, we will see a statistical significance. In summary, in cognitively normal subjects, we saw the clinical periodontal disease associates with amyloid deposition in the brain. We also found that subgingival bacterial dysbiosis associates with low CSF A beta. In conclusion, while they was shown in the rodents, we show for the first time that periodontal disease through clinical and bacteria associates with amyloid pathology. Brain amyloidosis may be modifiable with periodontal treatment. I would like to thank my collaborators from Cornell Med, Dr. Moni De Leon, as well as our microbiologist, Deepak Saxena and Smruti, as well as my students. Thank you.
Okay. Thank you very much, both Drs. Nath and Dr. Kammer. So we have many questions, I'm happy to say. And Jules Levin, who's one of the co-founders of the workshop, was the first to enter a question. And so what it builds from a nice discussion we had yesterday about whether or not there truly is accelerated cognitive decline in people with HIV older than 65, and it's directed toward you, Dr. Nath. So what's your opinion about whether or not people with HIV are at greater risk for either accelerated or accentuated cognitive decline compared with the general population?
So, as Scott, as you're aware, a number of these studies were done at UCSD and also by Victor Velcor when he was at Hawaii and now at UCSF. And so those studies clearly show that there is a subpopulation greater than 65 years of age who do seem to have accelerated cognitive difficulties, although that may not be the only thing that's happening to them. There may be more systemic frailty and other things are happening at the same time. So I think there is a concern that the process is not it's not only not halted, but even accelerated in some of these individuals.
Okay, great, thank you. For Dr. Kammer, so I'm going to combine a few questions, but they center around the pathogenesis of the oral microbiome in systemic disease and central nervous system disease. And so one question focuses on Porphyromonas gingivalis, whether or not that finding was reported that a single species of bacteria could be the risk factor for Alzheimer's or cognitive impairment. And you addressed that in part, I think, in your talk, but just to come back to it. And then related also to the oral microbiome's role in disease, what the role of LPS and TLR4 signaling is in chronic inflammation and disease, because there are TLR4 drugs being developed, targeted drugs being developed. So just to summarize again, do you think a single bacteria like Porphyromonas gingivalis is causative, or do you think it's more complicated than that? And then LPS from both gut and oral bacteria and TLR4 signaling.
All right. I was thinking a lot about it, and one of the reasons I added the P. gingivalis slide is because of the interest it got in the literature and in the media. But as you could see in my data, I don't have P. gingivalis as a predominant bacteria that was a biomarker of the difference between amyloid positive and amyloid negative. But that's not surprising, and I am sure in the gut is the same. Periodontal disease is not an infection due to one bacteria, P. gingivalis. Is P. gingivalis important? Yes, but not all the people have P. gingivalis, or not all. And it's the dysbiosis. Dysbiosis is defined by not only the quantity, quality, but also what bacteria is doing physiologically. I don't expect my dysbiosis, I hope I don't have it, it's the same like your dysbiosis. It's gonna be different. It's gonna be vary from one person to another. So P. gingivalis, it's important. I don't think it tells the whole story. However, P. gingivalis, it's a historical bacteria, and also it's a keystone pathogen that makes other bacteria do certain things. So what was reported in the literature, very important, that inhibition of the gingipine, which is P. gingivalis virulent, led to delayed or decreased pathogenic features in animal models. And that's not surprising. P. gingivalis gingipine, if it's expressed, changes the dysbiosis in the mouth. So maybe that's the reason. The other one of the LPS, definitely LPS, part of the animal models is LPS from P. gingivalis and a lot of other LPS. Whether there is a difference in response in P. gingivalis LPS compared to other LPS, including from other bacteria, there is not enough research in the on the ligand for the microbe. There was animal study and showed some reversal. So yes, it plays a role.
Okay, thank you very much. Avi, the next couple of questions focus on TAT-based therapies and both the development and the safety of them. And so one question from Jules had to do with are they being developed and what sort of timeline is there? And another from Thomas Villa has to do with the lock and block strategy. Are there side effects and any contraindications you anticipate for that approach based on what you know so far?
So in our lab, we've been interested in the antisense technology, and so the advantage of antisense technology is that it's been used, in a number of other genetic disorders, and so it's fairly safe. There's a little bit of immune activation that can occur with some of these molecules, but really the side effect profile is very well established. The advantage of blocking viruses is that you always worry about off-target effects with the host genome, but here you can find unique sequences that are non-human because you're targeting a non-human pathogen. So it's a human pathogen, but non-human sequences. So the safety profile should be very good. I'm not too worried about that. I think it takes time to develop these things, and then do all the clinical trials and stuff. So it's somewhat of a rate limiting factor. But compared to small molecules, antisense I think holds a lot of promise. Blocking, you're blocking directly just at the HIV genome. And it shouldn't be affecting other functions within the cell. Now, delivery of these things, I guess, is another challenge, and that is, can you get it to the right cells that are infected? And so getting it into lymphoid tissues and stuff can be done, but I think that it could be a bit of a rate limiting step. With regards to corticostatin and stuff, I don't really have a lot of information. I understand there is concern about toxicity there, but I don't have all the details.
Okay, great, thank you. So I'm going to find a few other questions that basically have to do with treatment with regard to oral dental health as well. So the questions have to do with what is the treatment for periodontal disease, and how can we alter if these associations are biologically accurate. How do we alter that relationship between oral health and cognitive decline? And I'll add in a little element of my own, which is as an infectious disease physician, I know that sometimes people who are edentulous are not at risk for certain infections compared to people with severe periodontal disease. So if you have severe periodontal disease, is being edentulous better for your systemic health than having severe periodontal disease? So it comes under the umbrella of what do you do about if you have moderate to severe periodontal disease?
Yes, if you have periodontal disease, you need treatment. And the treatment that we have is quite effective, which is scaling and root planning, which it's known as deep cleaning, but it's not enough. That's the initial treatment. Then this following that, you change the microbial environment subgingival under the gum line, but that has to be maintained, and that's why your doctor asks you to go each 3 months for maintenance, which means to do whatever it needs to be done to maintain the healthy environment. So that's one of the treatment. Of course, there are advanced treatment surgical approaches when the patient doesn't respond to initial therapy, and depends from person to person. In terms of, by the way, I didn't present in my data, but what we found is that subjects who had frequent periodontal maintenance had lower amyloid accumulation in the brain than the ones had less frequent periodontal maintenance. But in order to find out the effect of periodontal disease, you have to do a clinical study, and you have to match to for the initial periodontal treatment for initial periodontal condition. In terms of tooth loss, there are many studies to show that it's associated to cognitive decline. There are two aspects when you do studies on tooth loss. First of all, why did you extract the tooth? Was that periodontal disease that was there for longer time? So it's long exposure to periodontal disease. On the other hand, you take it out. Did you remove that, or the damage that it did is irreversible, and you cannot change it? So that's another way. Tooth loss, it's also a you don't know, confounder. Diet is a confounder for tooth loss. The diet change, and I can tell you that it changes. One of the things in my study, when I mentioned about homogeneity of the population of the sample, was that all the people had teeth in their mouths. They were healthy. The same thing to avoid confounding. And also they were normal. One of the things you mentioned before, that in people with dementia, you are worried about oral hygiene, how they maintain. And this is a worry. There are two parts for it. If you have periodontal disease, it is important to maintain the oral hygiene, the appointments for the doctor, as I mentioned, the maintenance. But also, on the other hand, periodontal disease is not only the oral hygiene, it's all and the bacteria that it's there. It's how we respond, how our immune respond to the presence of bacteria.
Okay, thank you very much. So we have about 5 or 6 minutes left. Let's see here, 5 minutes. And so I'm going to, again, sort of combine a few questions, Dr. Nath, for you that are focused on pathogenesis and of HIV in the brain. And the questions have to do with, one, how do you think the pathology now is different than it was 10 or 20 years ago, when people generally progressed to more advanced disease and our therapies were not as effective? And what is the influence, again, of comorbidities, and specifically Sylvia Guillemey mentioned addictive drug use. And David Moore asked about COVID-19, if you think that COVID is going to have an effect on neuropathology if people get co-infected with people with HIV get infected with COVID. So again, how is pathogenesis different now, do you think? What's the influence of comorbid diseases, particularly addictive drug use, and what do you think about COVID-19?
So I think the pathophysiology is different. What we never saw in the pre-antiretroviral era was an adaptive immune response within the brain. Now, if you look at the pathology, because patients have elevated CD4 cell counts, you can actually find lymphocytes infiltrating in the meninges and in the brain. Some of these patients, so it's like an indolent form of an iris that is taking place. So, and there is, and that I think is a major difference and has not received the kind of attention that it deserves. And the second thing is about the comorbidities. Yes, drug abuse is important. In fact, in our own study, and I didn't point that out, when we looked at TAT levels in the CSF, it turns out that the cocaine drug abusers had higher levels compared to everybody else. So what it's really doing, our hypothesis is that cocaine opens up the blood-brain barrier because it causes the vasculitis, and that can lead to increased seeding of HIV within the brain. And if the viral load is high, then the opportunity of producing viral proteins also goes up in that regard. Lastly, with regards to COVID, we don't have enough co-infected individuals, so all I can do is speculate. And what I would speculate is that it's not the acute infection. There's no evidence that the virus really invades the brain. We've done about 30 some autopsies now on COVID infected individuals. We can't find COVID SARS within the brain. But what we do find is a lot of inflammation in the brain. A lot of macrophages are there, some T cells, largely CD8 cells. And there are a lot of these post-COVID syndromes that are occurring in these individuals that mimic chronic fatigue syndrome. So my feeling is that HIV already has an inflammation problem. With the post-COVID, added inflammatory problems, the two together cannot be good. So the long-term sequelae of the infection is really a bigger concern to me, although theoretical at the moment, because we don't have a cohort to evaluate. But if I were to predict, I think that would be the bigger issue.
It's interesting because for many reasons, but one I read, which was more of an opinion, but was based on some observations, was that the severe respiratory disease and the physiologic stress has a number of effects, but it also, apparently, according to this article, downregulates ACE2 in the brain. And so paradoxically, if you get, if that's correct, if people who get severe disease may be less vulnerable to encephalitis because ACE2 actually downregulates.
Yeah, so you're absolutely right. ACE2 is the receptor for the virus, but basal levels of ACE2 are very, very low in the brain to begin with. So, and we cannot find virus in the brain. So I'm not worried about, infection of the virus. And what I'm worried about is inflammation, and why the inflammatory cells are going in the brain when there's no virus is not entirely clear. So I don't know what the target of it is, but the fact that it's getting in there is bad news.
So very quickly, just want to mention a question. We don't have much time, but Brianne Olivieri-Mui just raises the question about other conditions that are common in people with HIV, mental health conditions, and what the role of TAT and the oral microbiome and periodontal disease are, and particularly depression, because people with HIV are more likely to have treatment-resistant depression, and it's been linked with inflammation as well. So as cognitive impairment has gotten milder, interest has grown in these other conditions as well, and they're obviously life-altering also to have severe depression or other mental health disorders. So are either of you also adding that to your panel of investigations to look at other conditions besides cognitive impairment?
So you bring up a very important point, Scott. You cannot look at the brain in its entirety without looking at mental health issues. And the pathophysiology of some of these things may not be all that different than looking at cognitive dysfunction. Elegant work coming out of UCSD shows that inflammation is important in driving depression. And work done here at NIH in non-HIV infected individuals also confirms that major depression that is resistant to treatment is often associated with inflammatory markers within the spinal fluid. So I think you're absolutely right that we really need to take into consideration that that at least in a subset of these individuals, depression is probably driven by inflammation, and we need to be able to try and see how we can make an impact there by another class of drugs that has traditionally not been used for treating the plaque.
Okay, well, thank you both for participating in the workshop. It's our 11th year, and it's always a real benefit to the workshop to have people of your stature come and speak with us. So thank you very much.
Well, thank you, Scott, and thank you to all the organizers for putting together this really fantastic session here. Really enjoyed the discussion, and thanks for moderating.
Thank you. So Goda has asked me to remind everyone that there will be an award ceremony at 11:25 Eastern. So that's about an hour and 20 minutes. Is that right, Goda? You can type me an answer in Zoom. So I want to make sure that I'm seeing that correctly. But yes, okay. So please plan to be there because there have really been some outstanding young investigators this year. So I'll close the session, and now we have a short break for until 10:15 Eastern. So I ate into your time a little bit, only about 8 minutes away. So thank you everyone for participating, and particularly for my colleagues here on the West Coast and on Mountain Time because it's sort of early. So thank you.